environmental phenotyping. And I want to say that the big thing that we're trying to do here is bridge the gaps between the lab and the world. So we can learn, we can approach different kinds of questions. Ag bioresearch, and Doug Buehler in particular, is very supportive of this, and he supports this particular workshop because their mission is to engage in innovation and leading edge research that combines scientific expertise across spectra. And that's what this workshop is about, and I hope they'll become very clear in a minute. Okay. So the agenda of the meeting. First, I want to tell you about what this whole project's about. We want to have a chance to show off the cool technologies and the work that's been going on both here and around the world. We have a subset, a small fraction of it out here, but I'm very proud of that. A lot of the people have been working on this, and I want to give them a chance. We want to give them a chance to toot their own horns and tell you what they're up to. But the real purpose here is to encourage you and the community here to get engaged, get access to this technology, and contribute to it. So we want to give you access. The whole point of this is to get access to the MSU community, these tools, to do new things. We want to understand what your ideas are, where you're going, and how this platform can help you, because that helps us. We want to lose control of it. What I mean by that, and be surprised, we want the community, we don't want to control this. The best ideas are out there in the field, out there in the world. We want our technology to go out there and catalyze new ideas, and we want to be surprised by what happens. We also want to start a dialogue about the future direction of how we're doing technologies here in the center, and so on, and get your input on that. But we also want to give away free stuff. I'll come to that. How many of you know about the free stuff? Okay, good, great. Okay, so you've seen the very nice schedule of events here. This is the overview, which is like philosophy and what we've done in a broad context. In the afternoon, there are these breakout sessions, and they're concurrent, which is always a problem. You don't know which ones to go to. Go to one that you think is interesting. You could change, right? But if you miss something and you want to follow up later, the people who are doing these things are going to be available all the time. Especially for you people at MSU, you can just come around and engage them. Also, there are displays and posters out there, and the people using and developing the devices are going to be out there attending these things. There are going to be demos of both all the new device, the beta prototype, the Coral spec, and other things that have come out of this. So please go there, and that's also lunch. So here are the ground rules. These are them. What I mean is, let's be interactive, ask questions, join in, interact, geek out, participate, join something, propose, collaborate, and follow up. It would be really great if something came out, if many things came out of this, that we are actually actionable, that we follow up and that new things happen. Okay, so the free stuff. First of all, there's lunch. There's a raffle. We're going to give away the new version to somebody without any constraints. But this thing is set up between the center and Ag Bioresearch to get the tools into the hands of innovative researchers here. So we're giving away the devices, support, and technology to make your project work. Okay? So there's a very simple form. It's a real proposal. It's like a page or page and a half, I'd say. And we're going to gather them. There's a deadline. It's very short. May 10th. And these are going to be looked over, judged by a group of eminent scientists, extension agents here at MSU. We decide how to distribute those to get biggest impact. So you can be a student, a postdoc, a full professor, an extension agent, an entrepreneur. All of those things are welcome. So follow up on that. Okay, I want to get on to the exciting part, but I thought I'd tell you a little bit background about what all this is, where this came from. My lab's in the plant research lab. And over the years, we've been developing instrumentation to solve certain scientific questions, particularly about photosynthesis in plants. And we've been trying to see how photosynthesis works inside living plants. To do this, you need new techniques, new technologies to do that. But along the way, we said, hey, these tools would be useful for a broader community. So that's how CAP, the Center for Advanced Algal and Plant Phenotyping, was started. And its goals are to develop and disseminate transformative phenotyping technologies, particularly for plants and algae, and to integrate various disciplines so that they can work together on solving big questions. To establish a community of researchers here at MSU who work together, catalyzed by this technology, and has broader impacts. Get this technology out into the world, particularly in the developing world, to try to solve big agricultural problems. And I hope you'll think that by the end of all this, that the photosync is a good example of how that's working. So the goal, how we try to solve problems in the CAP Center, we want to bridge gaps in the current technology that prevent us from answering certain kinds of questions. In some cases, those involve developing new methodologies, new technologies at the fundamental kind of electronics or sensors or biochemical sensors, things like that. But other cases, the big gaps are in decreasing barriers to access, use, and interpretation of results across different scales. And both of those things are going to be evident in the photosync platform. Okay. So here's how we go about doing this. First, we focus in on a specific scientific question, right? A scientific problem. We're focused on that. We're choosing a question that requires new technological approaches to solve, but we make the solutions to that so that they can be applied to more than one problem. The next level that's really important is to engage the community in the development, the validation, application, use of the technology. Because you can't solve these questions. It's very difficult to solve these questions in isolation. And that part, I think I want to emphasize a lot during it. And then we want to spin off these technologies in ways that make sense, that have big impact. So it could be a company, could be a nonprofit, could be open sourcing things. The idea is to decrease the barriers to access, interpretation, and use of the technologies. Okay, so CAP has developed a number of technologies. These are a few of them here, and we'll talk about we're going to focus on the ones that are associated with the photosync platform. But there are also ones here for imaging plants in simulated environments and reproducing environmental conditions for growing algae, things like that. There's one here for hyperdimensional analysis of protein complexes. All of these things are focused on answering big, broader questions that couldn't be answered otherwise. Okay, now we get to the fun part, I hope. And I'm going to define the question that we're going to try. You can define the question we're trying to approach with photosync in several ways. I'll try to go through them. This is one. How many of you get this? Okay, this is the big question. The genome, by the environment, by management, gets to the phenome, how the plant performs. Okay, so in other words, you have a certain variety or genotype. You want to know how it performs, but this is a very, very complex interaction here. Those interactions are very difficult to get at, and I'll describe why in a second. But that's not the whole problem. The problem is, once you have this, you have to get actionable knowledge out of those interactions so you can improve them. Eventually, you want to improve. You want to know how the plant works, but you want to know how to use that knowledge to improve it. And this is what we call a hyperdimensional problem. It's very difficult. Now, we in academia tend to do this. We try to make this hairy, big problem tractable. And we do that using controlled experiments, reductionist approach. And that's very, very effective at solving certain kinds of questions. But there's a trade-off. Here, in the controlled environment, your statistics works. You can vary things very carefully. It's very tractable, but it's oftentimes not relevant to what's really going on here. And out here is a big mess. So many interacting factors. It's very difficult to control it. In the past, this was really the best way to solve these questions. But now, with new kinds of technology that allows massive data collection, we can use an information approach or a big data approach where you collect lots and lots of data and the metadata, and then you can understand what's going on out here, take that knowledge, and reproduce it here. Okay, so one of the approaches we're trying to do here, okay, how do we get at that thing? What are the big gaps? We have lots of genomic information these days, but the phenotyping, understanding how the plant works in the environment is the biggest gap, perhaps, I think. It's probably the next revolution in plant sciences if we can understand that and make those connections. So phenotyping is a big challenge because you have to systematically explore all sorts of complex phenotypes under many, many conditions. I won't say more than that. Each phenotype is a different platform. They tend to be very costly, complex. You need to connect it to metadata. You need to probe complex interactions under multiple conditions. And what we really need to do to sort this out is to have the technology that allows you to measure it in different environments and bridge the gaps between these different kinds of data sets. So here's our approach, our big approach. We want to do two things. We want to bring the lab to the field. So we have the tools out there in the environment that we can do. We want to bring the lab. Once we know what's going on out here, we want to reproduce it in controlled conditions to apply all reductionist approaches. And then we need the analytics to pinpoint the causes and so forth to get insights and to improve the crop, management, and so on. So this is our big workflow. PhotosynQ is up here. It measures photosynthesis, other plant properties in the field. The other platforms I've been describing fall down here, where you take that information, you can reproduce the environmental conditions and study things in detail all the way down to the molecular level or to the genetic level, so you can guide breeding and so forth. Okay. So this is where we're going this time. We're going to take sophisticated technology and put it out in the world and see what we can get. Another perspective is this. How many of you are basic research scientists? Great. Let's take this approach. I want to know how photosynthesis works in a living plant. I've been studying that for a long time. Does it matter? Does anything I find in the lab actually matter out in the field? Does it affect? Is it right? Is this something you only see in the lab? Can I use it for practical purposes? Things like that. Okay, there are a whole bunch of questions big questions you can answer. This is what I'm going to talk about today. Are plants too conservative? If you let them go wild, if you reduce the amount of downregulation, you get a bigger plant, something like that. Does your cool hypothesis really make any difference? Can what you do in the lab help people out in the field? Can you make a better plant? Can you predict something useful? Can we answer this kind of question? It's very difficult unless you have, you can measure the real performance of the plant in the real world in a high throughput with the critical metadata and the analytics so you can see those connections. That's the point. So can we go and get this coverage both at the hyperlocal level, the global level, and in the lab and compare those results? Those are two perspectives on how to see what PhotosynQ can do. There's another one. And I'm going to go this way. These are the big ideas. And by the way, after me, Dan and Greg are going to talk, and they're going to cover some of this from their own perspective and in better ways, I imagine. The idea behind PhotosynQ are these. First is to bring the lab to the world in terms of the environment. Can we get the technology where you can measure important things in real environmental conditions and capture the metadata so you know what actually happened? Second is to bring the lab to to the world in terms of people. There are two parts to this. Lots of people around the world are very smart people, but they don't have the tools to do the kind of science that could move things forward. Second is, I want to do the research. There's no way I can do answer the kinds of big questions I want to know the answer to without a community of researchers around the world. We've got to solve those problems by bringing the technology, the access to the technology, to the world. Okay? So we have to engage the community. We also have to engage the community in terms of innovation. We don't know it here in the university what all the real problems are. When you get the technology out there, you really can learn what actually is going on. And then we need to close that cycle. We need to connect these measurements in a way to the broader context to get actionable knowledge, both scientific knowledge and actual knowledge. Can you use this to solve an actual problem? Okay, so what is it? So the PhotosynQ platform is actually composed of two kinds of things. First, cloud-based network, like a social network kind of thing. And Greg's going to talk about this in more detail because that's his baby. And it enables communities of researchers, extension agents, farmers, entrepreneurs to communicate science. So, for example, here's a project lead. This person could have an idea, put it in the cloud with the instructions and protocols, and people around the world can actually join in there and perform those experiments. The data can then flow back and forth, and you can discuss, and you can apply analytics and things like that. Sounds pretty vague, but we'll get to some specifics in a minute. Now, this part of the chain here, this is important. This part of the chain is also a huge bottleneck, and I talked about that, the access to appropriate tools. So you can plug into the platform a series of instruments. They can be ones we made, the ones you made, industry could make them. They can plug into the platform and enable researchers to make new kinds of measurements out in the field, in the lab, and so on. What's cool is that this project here can instruct the instrument how to perform. It can tell the researcher how to perform the experiment. That data is all collated, collected, sent back to the cloud with the metadata so that you have immediate results in context. So this is an enabling. One that I'm discussing here is called multi-spec, and we're going to spend a lot of time on that. Okay, here's an example, which I won't go into detail because I think Dan's going to get better examples, but here's Kelvin, our collaborator in Zambia. He wants to know how common beans perform, which ones perform best under combinations of stresses. He comes up with a plan, proposed plan. He puts a project online. A group of people, either in his university or around the world, can join in there, use this enabling technology, and you get data, which is geotagged, you know who did it when, so on, all the environment parameters, and here's actual results. And these are different varieties under these different stresses, and you can see statistically significant data that he is going to use to map QTLs. So the point I want to make here is that that what you get out of this is useful to Kelvin. It's enabled his research. And I think that's really critical because we can't ask people all around the world to make our measurements unless it's useful to them. And there are many cases where this is true. This cycle here enables him to make measurements he couldn't otherwise have made. But let's look at it from above. We now have sets of data from all over the world like this. It's big data. Some cases we've controlled what's happened. Some cases we haven't. We've let happen. But look, we can look at it and we can compare all these data sets and look at it from a big data perspective and ask different kinds of questions. And I'm going to present one to you at the very end. We'll come up with an algorithm that can predict yield or disease onset. Okay. So, all right. Yes, the platform is also open to APIs, so you can now make cross connections to other phenotypic, genotypic databases and so on, to make connections, let's say, to the genetic component of the response and so on. Okay, let's now focus in on the device itself and what it does. This is one of the devices here, the multi-spec. But the idea is to develop tools that solve problems that fit into a certain category in this case, those that can be disseminated to approach new kinds of problems on a broader scale. Am I doing so? Okay. So Greg is going to talk about that, about how we chose which kinds of measurements to make or incorporate in the particular device. Well, we want something that's inexpensive, but that's not a toy. It's a sophisticated piece of equipment that can answer certain kinds of questions, specific questions. We want to make this very inexpensive. We're not trying to monetize this technology early on. We want to make it completely open and accessible. Where we see the value in that is getting the data that nobody else could get, because now we have a broad community of users. So we can get it and have impact that way and get kind of at questions that you couldn't. So here's the big thing. The tools we designed for this multi-spec takes the capabilities of instruments costing 30 to 75K and boils them down into a package that costs hundreds of dollars, about 1% the cost. They collect multiple data sets all together. This is important, not just one thing, multiple things, because you need that to triangulate as to what's actually going on. It's very easy to use, it's guided use, and it's linked to this platform to allow it to communicate with other communities and other tools and so on. So, and you can also communicate with expertise, and there's built-in educational capacity or possibilities there. This is the instrument that Greg's going to talk a little bit about more, but I wanted to spend a little bit of time telling you what it does, what its design principles are, and yeah, okay. So first of all, we have to measure important things or interesting things. They have to be interpretable. They have to be high throughput. We're taking the capabilities of instruments that take minutes to many minutes per sample, and we're boiling them down, not only the instrument, but the methodologies we've developed to make all those measurements in less than 20 seconds. We came up with 20 seconds because if it was longer than 20 seconds, Dan said there would be a revolution in Malawi. For the people in the field, they would never make measurements that lasted longer than that. So that's the kind for us. You can make any length you want. You can measure days if you want. But we want to make all of our measurements for the field to be rapid enough so that they're feasible. Because we're trying to measure lots of things. So it has to be what I call feasible. It means that you have to be able to place it in different environments and have it work. It also has to be open, because we want the use of these tools to be expandable. We want people to have new ideas, add new features, to get new things. So, and also clonable. We want people to steal it and do new things with it. You can do this already. We'll describe how you do that. The other thing we want to do here is make this whole process responsive to the community, the needs of people. So what do we get out in the field? We want to incorporate that in our new ideas and designs. Okay, so that's the new device, which I'm very happy to say you can see out there actually working. Okay, version 1.0. The one you saw before was the beta prototype. We made a few hundred of these things and sent them out in the world. And based upon the results from this and the feedback, we have that. And I'll tell you a little bit about what these do. What do they do? Okay, first of all, you can collect key metadata. Light intensity, temperature, humidity, location, timestamp, geoposition, genotype by means of inputting survey questions or reading barcodes. Okay? I want to also say that anytime you make a measurement, we're validating it against the state-of-the-art instrument that you can buy commercially. So we know that they're what we say they are. You can also measure pigmentation, colors, absorbance, or reflectance spectroscopy, so you can get chlorophyll content, anthocyanin content. Those could be indicators of plant status at various levels. Interestingly, you can also from these get something related to the canopy density and structure. We put in a lot of photosynthesis measurements because I like photosynthesis, but I also think it's a very useful probe of the plant status. So all of the fluorescence parameters that you would measure with a panfluorimeter you can do, and more, because we've developed new ones that let you measure things rapidly and in different environments. This can be used to measure electron flow, assimilation, photorespiration, photoprotection, photo damage, all that kind of stuff. You can also measure photosystem 1 phenomena and electrochromic shift, these kinds of things. If you're really interested in these things, it can give you an idea of the metabolic connections between the light and dark reactions. Photosynthesis. The current version has a little infrared gas analyzer in it. So you can, in principle, measure assimilation and respiration. That was the intent. Nobody used it for that. Instead, they went out and measured different things with it, which we really loved. They measured respiration from soils or from seeds. And there'll be talk this afternoon about that, about those kinds of alternative applications for these things. The new one has a couple of really interesting new capabilities. One is it has a differential porometer, so you can measure stomatal conductance to get water use efficiency and things like that, leaf temperature, and so on. Now, whenever I show this stuff, people think, multi-spec and photosync are the same thing. And this is not the case. I want to emphasize, different things can plug into the platform and have been developed based on what has been done or what the interests are in the field. And there's going to be a session about this in the afternoon, about new applications. It's an open platform, so it's very easy to modify. You can reprogram it. You can change it. You can add complete new functionalities. Measure grains, algae, the leaf thickness, all kinds of bioassays, soil probes, coral bleaching, and diseases. And those are going to be covered this afternoon. You can also do the same thing at the other end and apply new methodologies. Like I mentioned before, we've been working on all sorts of new fluorescence analysis procedures that allow you to measure things in different ways. You can also take the other end and apply any kind of big data analytics that you want because the data is accessible. Okay. I want to give you just a little summary of a couple of things that have gone on, just to give you my perspective on what's been going on. Up to the point where we stop, and I think that you're going to talk more about what's the scope of things that have happened. But needless to say that over 18, people are using this in 18 countries, and by the way, all this data comes from the original beta multi-spec device. But it's been deployed to 18 countries. Over 1,000 people have signed up and using the thing. We shipped 200 devices, actually 300, but the first 100 were crap, so they're gone. There are lots and lots of projects. Most of these are independent. Some of these we know what's going on, but most of them we have no interaction with them. There are over 220,000 photosynthetic experiments or measurements that have been done. And if you go to the website, you can click on these and get an idea what the projects and things are. You'll find that quite a few of them are garbage, but there's some real wonderful things in there too. And people sign up, they measure stuff, but they don't follow through. But a lot of them do, and the real good ones are really good. Dan is going to talk about case studies and proofs of concept at various levels. I'm not going to say that. I want to mention that in order to make all this work, he's come up with some very innovative ways of interacting with people and organizing projects through the mobile phenotyping groups and entrepreneurs around the world, and a mechanism to share the devices so they can be best used. Here's another I was going to mention very briefly about the use of the device for early warning systems for diseases. But the one I really wanted to spend just a little bit of time on is this one, because it's near and dear to my heart. Here's the big question. Does photosynthesis control productivity? Can you measure something early on in the growth stage with this device using measurements you can get from this device that can predict yield? Yeah. So here's the idea. What's cool about it is that this is something we didn't expect that came out of looking at trials that were going on all over the world, but mostly in Africa and Malawi. These are the ones that Dan set up, and he's going to tell you the story about how this came about. But you say, if you look at all that data, is there anything in that data that can predict something as complex as yield? So we did a whole bunch of trials over there. And as the trials came in, you could see the data come in. And you can say, wow, that's cool because there are statistically significant differences between these different varieties, different conditions. But does it mean anything? So at the end of the growing season, we started getting, it took forever, but we started getting yield data. You get yield data and you say, let's look at that data and see if anything correlates. And this is the cool, this changed our life. This data right here changed our life. Changed our whole perspective. Because here it is. This is pigeon peas in Malawi on varieties and conditions. This is yield, and these are other parameters we measure with the device. We also measure environmental parameters. If you look at any one parameter, you can see there's a sort of correlation. This is a multivariate analysis, so you have to think about it a little bit. But anyhow, good correlations. These two here gave us the best. And I call them NP1 and NP2. They're new photosynthesis parameters. And they tell us something important. I'll get to that in just a second. But if you combine those, this is where things got really interesting. Because if you combine these parameters, you saw something that was very tight. In some cases, the R-squared value was above 8.8. We thought, that's great, but it's got to be a fluke. But the fact is that we start getting similar data on a whole variety of different crops around the world. And Dan may comment more on that. This is very exciting. Why? First of all, it means we're measuring something important. Just in itself, we're just not random noise. But right away, it has some social economic impact. You can predict the yield, even a little bit better than you can just by eyeballing it. That's worth a lot, both monetarily, but also for planning, needs and so on, markets, storage. It's possible you can imagine ways of using that information in management context. Can you turn that into actionable knowledge? Can you find out when they're wrong? Can you diagnose what it is then? And can you ameliorate that? Can you use it in breeding? And yield is a very complex trait, but here we have the ability to look in more detail and more conditions to see where things, is this a trait that can be bred for? Basically what I'm saying. And it can be used for constraining models for, let's say, climate change effects. So we're very excited about that. I don't want to finish up with one little set of data that is brand new. Sebastian brought it to me this week, and I want to give you an idea of what it looks like to, at first, agnostically look at 150,000 photosynthesis experiments around the globe. So take all the data that we've gotten, you kick out all the crap, and what you're left with are interesting things. And let's just look at it, because I think it's really exciting. So here's you can do things like this. These are histograms of photosynthetic parameters. Phi 2. This is the quantum efficiency of photosystem 2. You can see the range. I have no idea at this point what's what, right? But there's some interesting features here. Why are these two lobes? Otherwise there's this very sharp decrease here. Okay. Those are interesting. But now, they don't tell you that much, except for this edge here tells you something really important, that edge, but I won't go into that. What really gets interesting is when you start comparing different results, different parameters. So here we're looking at different photosynthetic processes. I'm not going to tell you exactly what they are right now. I'm going to say we're plot one photosynthetic process against the other. And what that allows you to do is triangulate and say, what's going on at the biophysical, biochemical level? How does one thing regulate the other? What's the range of these things? And you get these big clouds of points here. I want to focus in on this one. Remember I told you there was one parameter that was very, very good, the best predictor of yield? That's this. And you can see already something interesting. There's a big cloud of points, but you can see there's a, if you look carefully, there's almost a line that goes from point 8 to point 8. The theory of this, our theory of this, we've done a lot of work on now, is that when things are on that line, photosynthesis is well balanced. It could be going fast, it could be going slow, it could be under stress, highlight the light, but it's always on that line, except when it's out of balance, when something bad is happening. And I'm going to give you an example. So, okay, so how do you make sense out of this big data here? So let's take some specific cases where we do know what happened. In this case, this is a set of experiments in Uganda. It's a very simple experiment. There's a lot of cases we can apply this to. But in this case, we're looking at plants that have either been well-watered or droughted. Very simple thing. And now you can superimpose those behaviors on top of this big data set here. And you get clear differences. You can clearly see this. And you can see it early on. You can see different things. This is the one I was going to show you here. This is the well-watered one. It fits on the theoretical line. The red ones are stressed. And you can see some of them fall to the top of the line, above that line. Some fall in the bottom. This is the initial response to drought. And this is where they're really, really seriously screwed up. And this is why this parameter is so powerful and easy to measure. The other thing this does is allow us to go right directly to science and come up with mechanisms. Okay, so I'm going to stop here and just give you, move on to the next person, but I wanted to thank, this is supposed to scroll down. There are lots of people who are involved in this. And you're going to meet them all today. There's no way for me to thank all of them here. A lot of the leaders, but also everyone who's been involved in the use of the technology and around the world, and the funders as well. And thank you. Okay. Sorry? Are there any questions? Dan is going to be the, no?
Well, David, you're putting all of these together. Are you measuring more of the energy status, the proton density, you got, when you put everything together, you got a nice straight line, so something's happening.
Yeah, and actually, I didn't want to go into deep because I ran out of time. I've already went over time. That parameter tells, so here's why this is important. Light's coming down and hits that plant. And this is why photosynthesis is a good monitor of plant status, I think. Hitting that plant, and if that energy can be used, the plant wants to do photosynthesis with it, right? It wants to have high efficiency photosynthesis. The problem is if it can't do that properly, then reactive intermediates build up and they can harm the plant. They can kill it, induce all kinds of things. So what happens is that when those situations occur, there's a series of mechanisms that go in and activate a process that dumps that light energy. It wastes it on purpose. And so out there in full sunlight, 50 to 80% of the light energy is just wasted immediately. It could be more. Okay, so the question is that balance correct? Is it wasting more than it should or less than it should? When it falls on that line, it's perfect. In other words, when you reduce the efficiency of photochemistry, upregulate dissipation, photoprotective dissipation perfectly. And the surprising thing is that you find all over on all the plants we look at, it's almost always perfect, except when it's in trouble. So when it can no longer cope with that, that's what happens. Okay. Yes.
So David, really a lot of ideas going on here. I really like the major aspect of the project. So there's a lot of movement right now trying to do DIY and stuff. So what are the thoughts on enabling,
Absolutely. And where's Greg? Greg, you're gonna cover that in your spiel? It's
Okay, they find the pieces, they say, they can go to Amazon, say, I want to get this resistor, I want to get this war.
Yes.
They can just do some simple soldering, then here you are, you have a little more mess back.
two parts to that problem, to the question. Yes, they can download all of the specifications. It's all open. All the hardware we use is open. All the software we use is open. You can reprogram it. In principle, one can make one. It's not a matter of simple soldering, because as you'll see, we're using like the cutting edge technologies, miniaturization and things, so it's a bit difficult. But if someone had very tiny little hands and a lot of patience, you could do it. Or you could have it made. But in principle, yes. And people around the world could reproduce the workflow that we have and make them. But I'll let Greg address that in more detail in his, is that okay? Yeah.
I'll just say really quick that we're at sort of an odd time where citizen scientists moving from like quirky, cool, easy to make tools that aren't predictably useful to complex tools which will actually have an impact. And then move towards complex tools which will have an impact. You kind of have to change how you interact with citizen scientists. That's what I need to be able to get. It's not going to be, it's going to be a little bit different. But that's also true, in IT it's the same thing, right? IT has all kinds of stuff in it, but you don't try to build your own IT.
I'll add one more comment, because Steffi, Brian, and Ruby are giving a breakout session on educational aspects of this. You can take this device now and make a completely new thing out of it. You can reprogram it, jigger things around, and already make, it's a spectrophotometer, fluorometer, gas exchange, measurement, data collection system. You can add new things to it easily. So in principle, you can trade configurations, you can trade protocols, but you can also modify them. Yeah. But I think it's going to become clear, and you're next, right, Dan? Okay, great, thanks.